Hi, Bio30. Today we're going to look at the first part of chapter 16.3 on meiosis, and specifically that is how the body makes sperm cells and egg cells, doing a type of division that will end up with a half number of chromosomes for those gametes, for the sperm and egg cells. But to begin with, I'd like to do a quick review of mitosis that you saw in the last lesson, just so we're clear on the 4 stages of mitosis and how this does involve a cell in the body. I'm going to talk about maybe a cell that's got 6 chromosomes in some little fly or something, a 6 chromosome cell, and how the prophase, metaphase, anaphase, and telophase stages happen so that we end up with 2 of those cells. So in prophase, we have those 6 chromosomes visible. We have 2 of each, 2 number 1s, 2 number 2s, 2 number 3s. And in prophase, we see that the cell membrane is disappearing and those chromosomes are actually dark and visible. In metaphase, all of those 6 chromosomes in this case will line up down the center, the metaphase plate, and you can remember they meet in the middle in metaphase. And in anaphase, the little centrioles are going to grab the sister chromatids and pull them apart. So all 6 chromosomes get tugged at that centromere and they get pulled apart. So you can see we're headed towards telophase, where we're going to form 2 cells and each cell will have its 6 chromosomes. So what happens here is this will still, as it does cytokinesis and totally pinches off, this is still going to be a 6 chromosome cell. And yes, it's a split X-shaped chromosome, but it still has all of the genes in each chromosome. So each of these are a 6 chromosome cell. And in mitosis, this is what we're taking is 1 body cell and splitting it into 2 body cells, and which works because of that chromosome that has doubled, and that has happened in the interphase, S-phase section of interphase, where we've doubled the chromosomes, and so that makes it possible to make 2 resulting cells after mitosis. So what I want to do is go back to meiosis right now and start our lesson on how the body makes gametes, which you know are going to have only half the number of chromosomes, so that those gametes can get together and form a zygote. So remember that you need a haploid. In this case, it's got 3 chromosomes. You need haploid gametes to make a diploid zygote. And so what we're looking at is how do we get haploid gametes made in the testes and ovaries. So mitosis, remember, happens in all of your body cells, but meiosis is only making the cell division that makes gametes. So the only place it happens is in the testes and the ovaries, the only location in the body that meiosis ever happens. And what happens is our resulting cells have half the number of chromosomes, but also an interesting thing is possible, and that is that we can have some mixing of the genes, and I'll show you how that happens. And so we end up, after mixing some genes, so chromosome 6 in a gamete, for example, can have some genes from the mom and the dad in that chromosome 6. So there's a real mixing that happens. So there's a unique gamete. Each sperm cell is different from the next one. Each egg cell is different from the next one. And we'll find out how that happens shortly. So in meiosis, when you have a chromosome with a cell with its diploid chromosome number, what happens is we have 2 stages of division. Meiosis 1, where those chromosomes actually, instead of being in their pairs, 2 number 1s, 2 number 2s, 2 number 3s, 2 number 4s, at the first division here, we actually will have the pair actually split up. And so as you see the sequence of events happening and unfolding here, you'll see that the pairs of chromosomes get split, and that's why in meiosis 1 we already have a half number. And what happens in meiosis 2 simply is that each chromosome gets pulled apart. And so what you end up happening is you're getting haploid gametes in the end, which are still a half number, and that's perfect for sperm cells and egg cells. So we're going to follow these stages through the next couple slides here, and you can check out this picture and how it's showing you, as you can zoom in on it, showing you actual microscope images of these cells going through meiosis 1 and 2. The lighter orange here on the top here, this is meiosis 1, and at the bottom the dark orange here is meiosis 2. And I like the color coding in here where you can see that a cell, remember this is only in the testes and ovaries, but a cell in prophase 1, the big thing going on here is that the homologous pairs, the two ones, the two twos, are going to find each other. And actually a very cool thing happens only in prophase 1, and that is that they twist around each other, and they might do some crossing over, meaning a chunk of mom's number 2 may swap with a chunk of dad's number 2, and the identical genes, the alleles, will swap places, so that what we can end up seeing is that chromosome from maybe dad's was the blue one and mom's was the yellow one, they may have chunks of each other's genes in there, giving this a unique combination for, let's say, a sperm ending up with this one strand. It'll have genes from both mom and dad. So that's kind of a cool thing that happens only in prophase 1, that we have the homologous pairs coming close. It's called synapsis when they come close, and crossing over when they twist around and swap some identical genes. So in metaphase, for the first stage of meiosis, we will see the homologous pairs line up together down the center. So they're not in single file like mitosis. They are found with their homologous pairs. In anaphase, those pairs separate, but it's the spindle that grabs the full X-shaped chromosome. So the two number 1s get pulled apart and the two number 2s get pulled apart, but it's the full X. So in telophase, what you end up having is only here two full chromosomes, but they're still in the shape of an X. If this was a human cell, it would be 23 full chromosomes and 23 full chromosomes. But remember, when we lined up, it's not necessarily mom's that was all on the one side and dad's all on the other side. That's not very likely to happen. So it's random which one is on one side and which one is on the other side. So when we get pulled apart, this is a combination of some of these are number, like maybe mums are number 1, 4, 6, 10, 14, 16, and the other numbers on this side are from dad. So it's already a very random collection of the 23 chromosomes. So in meiosis 1, we end up with a half number of chromosomes still. All of them are there, 1 to 23, but they're full X's. All that happens in meiosis 2 is that we see those X-shaped chromosomes. There's 23 of them on each side in human terms in prophase. In metaphase 2, we line up that 23 chromosomes down the center, and in anaphase, we pull apart the sister chromatids. We pull them apart, and so we still get 23 strands here on each of the cells that result. So you end up, in human terms, 23 chromosomes in each of the resulting cells. So this would be like 4 little sperm cells that result from an original one cell that was found in the testes. So remember, we are separating our homologous pairs. We are then separating the X's, and you end up with 4 resulting gametes made from 1 original cell. So the notes that follow walk through this. They mention here that interphase does happen before we ever begin meiosis, but it does not happen between meiosis 1 and 2. It just directly goes interphase, meiosis 1, directly into meiosis 2. And in interphase, remember that every chromosome from dad, from mom, does double, and then they're all in the shape of an X. They're doubled and ready to do cell division. In meiosis 1, then, remember there's this crossing over that happens in prophase 1. The pairs line up in homologous pairs metaphase 1. Those pairs separate with the spindle taking the entire X, and you end up with haploid. So if this was a 4 chromosome cell, you end up with, a haploid cell at the end of the first meiosis. And as you can see here, all that we have to do yet is to split those X's in half, and that's how we end up with actual 4 of them at the end of meiosis 2. So prophase, just to review here, synapsis is when they get close together. Remember in the nervous system we talked about a synapse being a space. Well, synapsis is closing that space and just getting close together in order to do the crossing over. When you have the homologous pair together, like 2 number 7s together, because there's 4 copies right there of all of the same kind of genes, that's why we can call it a tetrad. And you may see that word used sometimes. Okay, so crossing over, if we have the gene for, insulin that was on dad's number 7, it may have swapped with mom's information on making insulin, and they would have swapped places. And so it is in crossing over the exact gene that does switch places. This is a cool electron microscope picture of homologous pairs, and they've stained them. One is yellow, one is red, and you can see that when this is prophase 1, they got together and they have twisted, and they're crossing over in many, many spots along those 2 chromosomes. Pretty cool stuff. Metaphase, they line up in the center with their homologous pairs, and spindles are starting to reach out. Anaphase, the spindles grab on and grab the whole chromosome and move it over. Green doesn't seem to be the color to do here. Telophase, when we pinch off, we're going to end up with haploid cells, but they have the full X still there. Moving on to meiosis 2, we take our resulting haploid cell, and we have those chromosomes, full chromosomes, lining up down the center in metaphase, getting pulled now at the centromere in anaphase. And for each cell that went in to meiosis 2, we end up with 2 resulting cells. And because we had 2 cells resulting from meiosis 1, meiosis 2 will end up with 4 little sperm cells at the end of meiosis 2. Here is the kind of review of all the events of meiosis 2 that I've explained. And at the end, all 4 daughter cells are haploid. And you guys can take a look at some of the video links that are posted here, particularly these. And what I thought I would do is flip over to an image that you all have on page 29 of the notes that you can take a look at. And I just wanted to go through the comparison then of mitosis and meiosis. And so for starters, when you have page 29 in front of you, you can make some notes on the difference here. I'm just separating, for starters, this line on two sides of this page for the reason that that on the right side, we're talking about what happens in your somatic or body cells. And this is where mitosis will take some body cell that's, in this case, it's got 4 chromosomes. In human terms, it would have 46. And in the end, it's going to make 2 body cells each that are still haploid. And in human terms, that would be like making 2 more body cells that have 46 chromosomes. So recall from our first lesson that these PMAT stages are where the chromosomes in prophase are visible. You see the 2 longer ones are number 1s, 2 shorter ones are number 2s. In metaphase, they line up single file, and in anaphase, they're pulled apart at the centromere, and in telophase, they will pinch off, cleavage here will happen, and you end up with 4 strands on each side, giving you 2 cells with the original number of chromosomes as we started off with. To review my meiosis, though, on the other side, I want to just point out that this is, you guys can label those in, but this is prophase 1, metaphase 1, anaphase 1, and telophase 1. On the other side would be prophase 2, metaphase 2, anaphase 2, and telophase 2. And the significant thing over here is that if you ever get a question about where crossing over happens, it's only ever going to happen at prophase 1, where you see these, in this case, 4 chromosomes, and the homologous pair, 2 number 1s, 2 number 2s, are crossing over and swapping their genes. If this was a human cell, we'd start off here with 46 chromosomes. And what's happening here in metaphase is that the homologous chromosomes line up with their pair at the metaphase plate, or in the center of the cell. And in anaphase, the spindles pull apart and grab the whole chromosome and pull it aside. So in the end here at telophase, you will end up now with an N equals 2. There's only 2 full chromosomes, but 1 of each. In human terms, this is where you'd already have 23 chromosomes in each side, in each cell. So the very significant event here is that our homologous pairs, the 2 number 1s and 2 number 2s, 2 number 3s, separate. Okay, and what's going to happen in meiosis 2, the event, the major event here is that it's going to be the sister chromatids that separate. And we start off in meiosis 2 seeing that we have only 2 chromosomes on each side, and in human terms it would be already 23 chromosomes. What they do in metaphase 2 is line up single file, the full-on chromosome. In anaphase 2, those spindles come and pull those chromosomes apart. So they were looking like this, and the spindle pulled them apart. So in the end, you have 2 here, 2 here, 2 here, and 2 here. So we can draw tails on these. This would be indicating that this would be like 4 sperm that are haploid. And if we were talking human terms, all of those would be 23 chromosome sperm. Make some notes on this. Screenshot it if you like. Take that and check out your page 29 and watch some videos, and we will have a chance to go through this again in our meet on next week, scheduled for, I believe, on Wednesday, when you'll have a chance to check out mitosis, meiosis, and we can review